So hello everybody. Still quite a few in the audience, although it's Sunday afternoon. I can say that this wouldn't happen ever in Sweden. So I think it's a very good sign. The end discussion, which I will chair, will take place with the following panelists. May I ask them to come up and sit here? President Bertil Andersson, are you here? Dr. Erik Betzig, Professor Stefan Hell, Professor Ram Sasikaran, Professor Barry Sharpless, and Professor Tom Brown, and also Professor Stephen Smith. So we have enjoyed a fantastic program here, and this end discussion I would suggest we do in the following way. We continue with some questions. We will have Korea with us for a while, and then some questions from the audience. And then I would like the panel, and that's why I say it now, because you could think a little about it. Rather than reflecting on the various technologies and tools that we have been discussing. Could we do something with the sound here? Okay. Try to look at the frontiers of upcoming health and medicine. Of course, medicine today, that's everything from diagnostic methods to therapeutics, but also preventive medicine. It's very difficult to give prognosis, especially about the future, and I don't expect you to come up with something fantastic, but I still think it would be good listening to all the advice to young people, how you should try to disconnect. So I have the hope now that this panel will be very creative. There are no limits at all. But let's start with the disconnect this one. Now I'm completely disconnected. So let's start with Korea. Do we have Korea here?
Yeah.
Fantastic.
Hello,
Still.
we're here.
What's time in Korea now?
5 o'clock, exactly.
Okay, so it's time.
1 hour later.
Yeah, so it's soon time to go home.
5 o'clock.
5 o'clock.
Okay. So could we have a question, please?
We have nothing to ask.
Not right now.
Okay.
Not right now.
Yeah. We take that as a sign that this meeting has been very informative. You have been very saturated. No more questions. Very good. Anybody in the audience? No youngsters, you are tired. Some elderly gentleman or lady here who may have a question? Okay. So if not, I suggest that we start the discussion. Where are we moving? If we try to look, say, 10 years ahead, that's not a long time, you may say, but if I go back 10, 20 years, I saw a movie with somebody having a phone in the car, making telephone from the car, and I thought it was very futuristic. We know now that everybody has mobile phones running on the... You see sometimes people going in the street speaking to themselves. When I was young, when somebody was speaking in the street, well, you know that it was a mad person. My grandmother was head of a big mental hospital, so I saw many of those. Where will we be in the future, Barry? 10 years ahead?
Hard. Future, is it working? Yeah. some things are hard to predict, especially the future or something. But I think back to, people should, I guess they have to think back, you know, this back. Because, like, Polaroid's gone. Kodak wasn't gone for a long time, but I was thinking back, but it's gone now, and it had to go. and things like where you can make things effectively, they move. And so the things that sustain us in this world are always changing, and we know that, but we don't want to operate on it. And I guess the click chemistry thing was like Back to the Future, and I finally got the money from Richard to do it for 10 years with nothing to show. Then we got lucky, which is what you usually do when you find something. And Eric's thing, that I really missed some of the best talks that I think this afternoon. I caught but these things that I heard about them, and yours, I have to talk to you if I can, because this business of coming back, people think if they take 2 years off and they're going down the hill and they're gone. It had nothing to do with that. If you have intense creativity, and especially if you have a need to solve a puzzle. And one of the things I notice, things come back. It's iterative thinking. It's almost like you're a boring person. Your mind is boring. Why is it coming back to me? Most people put down, they go 2 steps, there's a wall, okay, that's there. I think that's what, if you don't have that, I'm not gentle. I once got to the counter going to Europe in the Continental thing in Newark, and I had to get something out and give it to the person, and Jan was standing there, I went, ah! and the woman said, bet he's fun at Christmas time. But back to what, you know what I mean? If it comes back, it's telling you something. It's not just you thinking, it's the subconscious mind, right? It's,
again, I usually think that...
You have to speak in the...
Trying to. There we go. That there's, I think a good scientist always has like 15 or 20 puzzles that are percolating in his brain, partially in his subconscious, that are not mature yet, and they're missing something. I mean, I know I have a number of ideas that just aren't practical right now, and they're waiting on, just like seeing that you can photoactivate a protein was the missing link in that one case. That I think that's what you immerse yourself in problems, right? And then, like you say, no matter what you're doing in life, you can be doing something totally different. The problems don't go away. They don't go away from your thoughts. And when, you know, you learn about something that's the key to open that lock, you know, you just have to pursue it.
But there's a famous saying, is that mediocrity imitates, genius steals. And, you know, that's certainly true, because it's not a matter of stealing in the bad sense. It's just this comes from there, and then that comes from there, and then right under your nose is that thing all the time. But when it comes time to see it, another weird thing is how it tends to run around the world sometimes. Everybody sees it at the same time. That's kind of a collective subconscious thing, right? And but when you...
If I return to, both you and Eric spoke about the importance of sort of being disconnected. I like that. Otherwise you hear everybody seeing the same thing, the same observation, they're going on the same bandwagon. That's no good. Steve, now NTU will go into medicine much more seriously than before. And we have heard from the questions of the young audience here. By the way, I should say that I think many of these questions have been very original, very creative. And we are not here to teach you. You could tell us a lot, I think. And as I see it, if we look at the Nobel Prizes, you know that they have mostly been to physiology. It's medicine or physiology, but physiology, that's basics of medicine, is much easier to cut good things quickly, and I see it. I'm not a medical doctor. We have Erling here, who probably better than me to answer this. I see it as a problem that there is such a gap between clinics and the fundamental science. So I think it's very important, it's instrumental that the development that occurs on basic level in biology and molecular biology could handshake with the clinics. Do you have any comment on this? This is NTU. No, sorry.
The first thing to say is that the gap between the clinic and the basic science should not be as great as it is. You've heard fantastic 2 days of the extraordinary developments in molecular science in its broadest context. The first is that you have to have a cadre of clinicians who are trained as scientists, otherwise in the States known as clinician scientists. And unfortunately, it is only they. I'm a clinician scientist. I trained with the MRC and in the United Kingdom, but I'm also was a fully practicing clinician. Mm-hmm. The mistake is to think that a jobbing doctor who works all hours God gives him or her and is interested in just treating patients, it's a complete mistake to think that individual can communicate at the same molecular level and the same scientific understanding of the disease, whereas a clinician scientist is trained in the molecular basis of disease. And at the end of the day, what you're trying to do is to match up the molecular understanding of the disease and the response to surgery and or drugs with the basic science. And that's the crucial link which needs to go. The microscopy that we were discussing earlier on, fantastic stuff, but you have to be able to put that into a clinical context. So the first bridge is to have proper clinician scientists. So one of the things our school will do is from the very beginning, it will try and bring in the concept that science and engineering and technology is a fundamental part of being a modern doctor. It is not an optional add-on. It's what defines the modern medical profession in 10 or 20 years' time.
If we go to, I thought I should follow the round here. It's simpler, and then you get a democratic possibility to influence. So next guy is Tom here. And to you, I will give the ball that we have many possibilities to develop diagnostic methods as long as we are in the lab and in the test tube. Everything is much more easy, and it's just a matter of if the sensitivity. We have heard the PCR, for instance, what a revolution it has had, but there is also a post-PCR area where other methods come in and become very important.
The one thing I would say is that the difficulty is that often in the practical sense, fancy tests don't work because they're too expensive and they don't actually make a substantial difference to the disease. Sometimes they make a profound change. So the issue with the application in medicine is that not only that, but you have to persuade politicians to pay for the extra costs. And it isn't simply a matter of scientists or doctors saying this is a fabulous fancy new test. If it costs 10 times as much, it's very difficult to get it in. So putting it into that value-added difference is, I'm afraid, as we both know, a boring but essential part of the application process.
Not to speak about safety. If we take modern technology, the latest in IT, the aviation would love to implement it on their planes, but it's a very long road. The computers they have today, which is normally around 10 computers per plane, they were sort of developed 20 years ago.
Yeah.
I mean, on the same kind of context, we all know now that there are very, very cheap methods of sequencing DNA, and obviously that brings us into an interesting area in terms of using drugs which have previously been not usable because they produce side effects in 1 in every 10,000 people, which could be fatal. But now we're beginning to understand the genotypes of the people that will have this problem. Soon, I think what will arrive is cheap machines. So not only will the actual few dollars to sequence DNA, but the machines will be cheap. That you can see in 10 years' time that you will have in your supermarket. Right? Your supermarket, you will have your pharmacy, and in your pharmacy you will have these machines and some of the drugs which are now prescription drugs, maybe statins, for example, will be available across the counter, and you'll be able to get your 5-minute test while you go and buy your soap powder and your other groceries, come back and get the drug over the counter. This is actively being pursued at the moment. But the impact of sequencing, diagnostics, and also in a different area, the impact of pharmacogenomics, whereby you can look at the progression of a disease and you can change the therapy to suit the disease more precisely, is going to have a massive impact for diseases like cancer, which change. I mean, the cancer changes, the treatment needs to change, the success rate will increase. So extremely cheap, high-throughput DNA sequencing with cheap machines, I think, is going to have a big impact.
And also involving IT, you may have personalized medicine. You have your genome sequence, you have some ID when you go into the drugstore that will tell you that you should not use statin, for instance.
And I think also that, the idea of sequencing a genome for $1,000, which is going to be, it's pretty much there anyway. It's going to go down and down and down, so you can have your genome sequenced every few years because it's going to change. It's not going to be the same genome that it was 5-10 years ago because we get older, and we get older, our DNA changes, our epigenetic, our epigenome changes. So, I mean, this is going to have a massive impact, but obviously the science that follows this to understand what these differences mean, that's going to generate a huge amount of new research. And I think if we come back in 10 years' time, we will be really amazed, as we always are, at how ignorant we were in 2012. Yeah. But that's kind of a general comment about the way that science goes.
As to methodology, both Stefan and Eric, you have really shown the importance of seeing. Seeing is understanding, and seeing life systems. I think you are mute. Yeah, you have to speak up. Or bang it in the floor, something. I think the blue microphone.
Good? Yeah. Okay. Because that's what we do. It was Helmholtz, who was a visionary guy, not just a thermodynamics expert. He had all kinds of ideas about everything, and sort of like the famous German before him that was doing the calculus. No, it was Leibniz. Yeah, this man was awesome. He saw that Cartesian thinking was a disaster. He was the first one to see that, and I think, but the business of seeing, he says to his students often, if your only evidence is visual, you're in trouble. You cannot. That's very shaky evidence. So that's our need, you see, to see. In fact, we all love it. We get thrilled seeing our movies and your movies, and we think that. But see, the function is what matters. Seeing it can be done as a wonderful journey of adventure in discovering what really happens, but the function is all that's needed. Yeah. And we think we can get it from seeing. My point is that to see, we always take things apart in funny ways, and who's going to put them back together again? You can do that with function right up front. Yeah. If you only watch function, but no, we got to know the mode of action, and this happened with an inkjet printer back in the 70s. This guy invented something at Stanford in Palo Alto, and his boss, he showed his boss one weekend, because he worked on weekend. He said, I haven't taken time out of the laser writer, but I got this inkjet printer idea, and it worked great, but he said, how does it work? Why does it do that? It spits the ink, right? Yeah. He didn't know. It was the piezoelectric effect. Yeah. And, he said, we can't touch it, And then Big Boss said the same thing. Namely, hey, you're not going to kill anybody with this thing. It works. This wasn't understandable to me. I see it everywhere. It's the default thing, right? If you want to have understanding and pretty pictures, we're going to continue to do that. That's what gets people's attention. That's why Ertl's work was so wonderful. You could see those waves, those dissipation waves going, right? Sorry.
Okay.
Say that.
Yeah.
So still seeing,
No, I
could,
think-
seeing could help us to imagine and
Absolutely,
continue.
yeah.
Yeah.
Because the eye, of course, and the brain is very good at seeing and perceiving the great picture once you have, say, all the data in parallel. But I think it's also very important to keep in mind that the images are quantitative data that show something, that really mean information, high level of information, as we've heard today, down to the molecular scale, down to the nanometer scale. So in that sense, I believe that by seeing the molecules at that scale, we learn something about function and functionality, and we can extract out of this quantitative data, which was not accessible before, function. But the point I would like to make here is the following. We've seen that we can see sharper, say, image the nanoscale with focused visible light. But I don't think that's the end of the story. I think it's just the beginning. What we will be able to do in the future, in my view, is we will be able to control the nanoscale. So in essence, the nanoscale is now being opened up for focused visible light. That is something sort of heretic. Maybe 20 years back, everyone had said, how would you access the nanoscale with focused light? Because there's diffraction. How can you control something at the nanoscale with focused visible light? No, I think we will, in the future, also be able to control things using focused visible light by using the molecules. And I give an example of what I mean. Many of you, or almost all of you, are familiar with optogenetics. You can control optically, for example, the neuronal action, for example. Now, you can imagine playing the on-off game, as we've seen here, at a much smaller scale by shutting off some of the, say, effector molecules in a certain region, but leave only in a very small place them on. And so you can control things going on at the nanoscale with focused visible light. That means you can penetrate into something transparent, something living perhaps, and at a very small scale determine what the molecule is supposed to do. Unimaginable a couple of years back or 10 years back. So this is enormous potential. And of course, I mentioned towards the end that you can build in principle any structure in 3D in a transparent sample. People in the past said, okay, for doing that at a nanoscale, you have to have X-rays or electron beams, but that's clearly not viable for a fundamental physical reason, because you can't focus the X-ray that well. But now we can build up, at least in the concept, say, molecular arrangement using focused visible light in 3D. I think this is a fascinating prospect, just by having two states, separate states. One does something, the other one does nothing. And this is, if you think about it, very simple. It's very simple. Because it's simple, it's profound. It gives such vast array of opportunities. We're just at the beginning, and I'm encouraging, say, the young people here, think about it. Imagine your own, say, invention or something that could come up simply by playing with the states of the molecules. You need just two states to change things at a nanoscale. And there's something else I would like to mention, because you mentioned being disconnected. I can just a bit about my history. So I did my master's work and PhD work with a solid-state physicist who had no idea about optics. And he wanted me to work on an optics problem because he was interested in making lithography structure with optics and inspecting them. I got bored in my PhD thesis. In fact, I felt that it would go down the drain. And I was terribly worried. I was thinking, oh, I'm now supposed to work with this 19th century technology called optical microscopy. There's nothing I can do. I will fail. And this started to make me think, what else is left in microscopy? Is there any fundamental problem that I could solve? And the only thing that was left, in my view at that time, was the spatial resolution. And this situation of being frustrated and worrying that my thesis may go down the drain made me creative and think about the problem. And so I think it's, again, very important to be disconnected, to see things from a different angle, not being part of the community. If I had been part of the optics community, of the spectroscopy community, all the people would have told me, oh, this will fail, this will fail, this will fail, this will fail. In fact, people did so. They said, oh, the molecule will not do that, and you will excite the molecule, and all these arguments. But since I had to look at things with fresh eyes, I had the courage of doing it. So this is very, very important, to have some distance and not to be misled by previous knowledge. Think out of the box. Yeah. Just don't be misled by previous knowledge. You should know things and be attentive to everything that is being said, but have the courage of seeing things with completely different eyes.
An alternative way is to be a little ignorant. That helps.
To some extent.
Not studying the literature too well, or misunderstanding it.
overinterpreting it. Yeah.
Eric, what do you think? We are speaking about seeing sharper and sharper. Is that really important? Maybe it's more important to see motion.
Well, it's like I said in my talk with the triangle. I mean, you can gain information anywhere. So I'll say a few things. First, from what Barry was saying about the double-edged sword of imaging, is that I totally agree with that, is that imaging is a tool, okay? But it's one tool among many. And it's like the parable of the blind men trying to see the elephant. And so, you know, we're looking at the trunk with imaging, but it's not the only tool. I mean, I work under the postulate that imaging is a particularly nice tool because it can be dynamic, exactly. Exactly, and then it's connected to function. So that's one of the reasons, again, that I kind of moved away a little bit from the super resolution, is because it's such a challenge to do a lot of dynamics with that. And yet, you know, the dynamics, you know, like Scott said, I mean, that was what really motivated me, is that part. The other problem that we face, though, with imaging, the caveat that you have to remember, is that the image is a mapping from what the reality is. And there are several steps of translation between the reality of the sample and the image that you get. And those steps are what kind of sample preparation we've done to it, how has the light affected the sample, you know, what is the limitations of the microscope itself that's doing it. So we should never forget, and I think this is going to be a big problem with the new microscopes, is how people overinterpret what they see in these images, and they got to remember that it's a mapping, and that if you don't do the right controls, the map will mislead you from the reality.
So what we actually image is the fluorophore distribution, not the molecule that is attached to it. We image the distribution of the fluorophore, but that's the job of the microscope, to image just that. And of course, the labeling will decide to which extent, of course, the fluorophore mimics the object that we look at. Of course, we have to keep this in mind. This is essential.
Something we haven't addressed here is the solid state, crystallography. You know, for proteins, that is the source of information, how the atoms are placed in space. But it is showing it for a completely static system at equilibrium, a dead system. And furthermore, the resolution today, although it's 2.5 angstrom or so, it's often not enough to get the really fine details about the chemical reactions that occur. We spoke about the F1 ATP synthase earlier today. We fight with this enzyme today. And because it's unclear if there is a water or not at a certain position, we cannot really grasp the catalytic mechanism. There are a number of really elusive things coming up. And solving a structure, which is the only structure you can solve, is like the guy who had lost his car keys, and the policeman came and asked him if he lost him there under the streetlight. And he said, but I lost it there in the dark, and there is no meaning to look there. This is something I think we have to keep in mind.
And another cautionary tale with crystallography is that, again, you're extracting this protein from its native environment. We got a guy at Janelia, Tamir Gorin, who does cryo-EM on it and sees how important the lipids surrounding the protein are to the conformation and structure of the protein. And so, again, it's this mapping problem that things get perturbed.
Still, this is how the big pharma factories work. They look for the small molecules, how they can dock into the proteins. So if we come to the big factories here, Ram, the sequencing factories, what will come out of it? Can you see the forest for the trees?
Minus 300 degrees. Is that right? I mean, is it minus 300 degrees centigrade? What kind of work is it? I mean, this thing's nearly frozen down to life. I mean, you know, it's one formula, like a football field. The first inch of the football field is all you're seeing. This thing is like a flying spaghetti monster. They come apart, they show you bubbles. I imagine them just crashing around. It's really cool what they do. And I also think that...
You have to take the microphone, I think. It seems that you have something important to say.
something moving, or see something like that's what life is. Eshimoser explained it one night to me in a long car trip. He's not a catalyst chemist, but he says there is nothing alive about a live thing unless it's dissipating through time, energy. It's just a bunch of things that can't be stopped. You stop them, that's death, and everything's dead. If that's such an appropriate thing, it's all the same material. But if you can see something happening and you can watch it, well, somebody does a simple thing to it, add 3 inorganic minerals to the system. I mean, you could find amazing things possibly, right? That would be non-toxic, would be cheap, and it wouldn't be a singularity, no silver bullets. You could watch anything that'll give you the function. The animal stops having the infection, the breathing, or in electronics, they want a better transducer of light, and it's the function we want. So if you had a way to screen, it sounds boring to people. It sounds like industry or something, but it's different. It's actually power. That's what life's lesson is. It doesn't ask questions. It only transduces more energy. If it doesn't, we'd be boiled out of here already, like Venus probably, you know? And it eats the energy, and it eats more. If you eat energy, you get more. That's an odd situation. That's life is eating everything here that can be eaten to make cellulose mainly. And termites are the #1 body mass on the earth. One of my polymath students told me that. So I thought, oh, that's interesting, but it's sort of obvious, isn't it? Otherwise we'd underwater under trees. So anyway, I think like your system, if we could watch, if those things started going some other way, we need to grab it with something cheap. I think you're talking about diagnostics, and I agree, that's really cool. Do you know Ioannidis at Stanford? He used to be in Athens. He's got an article that's written about him, and he didn't have a platform, but he was a surgeon at Stanford. He went back to Greece. He started getting interest in numbers and analysis statistics, and he got a platform when he came to Stanford. People started listening to him. But he says 2/3 of all medicines they're not significantly, there's nothing there. And we're doing better today because the water's clean, and most of us are healthy. This guy is really interesting. He doesn't get excited. He just has the data. And I have the article in here if you want to read it. I thought, you said no one here is at Stanford, right? But he has a much...
crystallographers.
Yeah,
right?
okay.
Okay. So
I'd like to comment.
Yeah, okay, yes.
Okay. So I like to build a theme on, I think the question that you asked is what the next 10 years might look like in a broader sense. And given the theme is molecular, I'd like to put that in context.
Could there be anything else?
No, I see the spirit behind that question. And the theme I'd like to build on is on the practice of medicine. And particularly given that several of our understanding in terms of the molecular systems essentially has very important implications in not only understanding medicine, but practice of medicine. And if you look at how medicine is practiced, it's fairly discontinuous and episodic. We often treat things that have a problem. And the analogy that I like to draw is that of the automobile industry. 20 to 25 years ago, in that time frame, it was fairly routine to change engines of a car and have major repairs on a car. Any automobile was routine. It was not, it was okay to do major changes, and it was part of how cars were built. But if you really look at the automobile industry in the last 5 years, or even the last 10 years, cars are made to last. They have routine diagnosis. They generally run much longer. And in part, what has transformed that industry is how the industry has seen this to be fairly important as not only a paradigm shift of automobiles that are made to last for a longer time, but as a consequence of how diagnosis, test, and different things are done in a fairly routine way, so that it was a combination of a mindset and bringing technology platforms. So what I envision, and as much as I completely agree there's a challenge in the mindset currently, but with where our measurements are, whether it's genomics and the variety of different things that we have heard, including imaging, that the so-called episodic way of measuring things to a continuous way of measuring things would be the future of medicine, where the molecular principles, the quantitative nature of these types of measurements. I'm not suggesting in the next 10 years that we would solve the problems, but we would start going in the direction where the convergence of life sciences, engineering, and technology, as you pointed out, would drive or would be a catalyst, a critical catalyst in the way this could happen. Now, an extremely important ingredient in that would be the way we really hope that the younger generation would embrace a kind of a very different way of thinking about this. It's critical that we start to begin to appreciate what the possibilities are from the science that we are learning, from molecular biology to, to the variety of technologies that we heard in the last couple of days. The utility of that, no doubt, is to understand the fundamental science and getting to the core of a discipline. But the beauty of it, as I would see, that I think in the next 10, 15 years we would see the transformation, is how we apply that in a pragmatic way, in the way we begin to think about medicine and hopefully how medicine will be practiced. I do know that's a long journey, but I think it's an important journey that we would undertake in the next 10 years.
And the question is if we can actively think in a strategic way. I don't think so. I think we will be subject to surprises all the time. And let me take one example. We have heard a lot about nucleic acids, and we work with them too. And when I started to be interested in DNA, people say, why do you look at that? That's chapter 1 in biochemistry textbooks. You should go for proteins, the engines of the cells, and so on. We have recently learned that there is something called small regulating RNA, small interference RNA, microRNA, and so on. These evolved due to that they were originally just the sewer from mistranscribed messenger RNA. They were just the remainder. And I think there is a general conclusion to be drawn that the cell hasn't. It may have all kinds of molecules that are wrong and that it has to get rid of. But these molecules are also used somehow in a regulatory fashion. There is not a single molecule that is not used. And the small RNA molecules forming a duplex of DNA that we heard Andy Fire understood was a duplex of DNA. That was due to an old discovery by Alex Rich in 1957. He showed that Francis Crick, who used to be right, he was actually wrong. He said, well, RNA cannot form a double helix because steric reasons and so on. And everybody, of course, believed him. But then Alex Rich showed that there could be a duplex. Now, the small RNAs, it has been resembled by a discovery of a beach, of a continent that remains to be investigated. And I don't know if that is to exaggerate, but this is the kind of surprises that will really confuse us and that we have to take position relative to. So something I thought of, and we pursue in my lab, is to look for the wrong thing in the wrong places. Because there is something called entropy that is shaking around things. There is always a probability that something that should be here leaks out and appears at a certain very low concentration at another place. And of course, in the textbooks, that will not appear because nobody has looked for it, nobody has discovered it. I postulate that if you look for the wrong things, let's say DNA in a plasma membrane, DNA should not be in a plasma membrane. If you do that, you will find it. I know it is there. Guschelbauer, 1972, said, it's not an impurity. I always found 0.5% RNA, I think it was, in the plasma membrane. So I think that rather than doing strategic thinking, we have to be, still in a strategic way, of course, be prepared for all kinds of surprises. We have to think in a very imaginative way and not be locked into paradigms, but which is, of course, impossible because, and you young people, you have your exams and so on, and there you have learned that you have to, a certain answer is right or wrong. And, but as a scientist, there is nothing like that, right? Bertil, as a president of this illustrious university, I'm sure you have visions for the future.
Absolutely, absolutely. Well, I think a very simple statement is that the molecular frontiers must contribute to our challenges. I think strategic or by surprise, it has to, at the end of the day, serve that. And Royal Society recently came out with sort of 10 grand challenges after a very broad consultations. But basically these two challenges are within health or sustainability, you could say. They seems to be. And I know you have a sustainability molecular frontiers in Stockholm in 1.5 months, so this is about health. So of course molecular frontiers as an organization is also well positioned, but this is what we expect, of course, from such an excellent, illustrious organization, yes. With a modest chairman. Yes. Now, since this was about health, I think, and you actually said, how will it look in 10 years? And then it's very useful to look, how was it 10 years back in time? And then the human genome had just come. And we know now what happened in these 10 years, and there's a lot of more data, more detailed data. I'm also, today maybe synthetic biology is where sequencing genomics were 10 years ago. So I predict that we're going to have a lot of more sequencing leading to personalized medicine. I mean, that is inevitable in all meetings I've been to. All the data is probably not going to be the challenge. That is how to revamp the healthcare system to that. Infrastructure, doctors, people who work at the hospital, and medical education. If I really would take this seriously, and I would say to Steve Smith here, you know, we have to do a completely revamped curriculum for our new medical school. So that would be a real thing, and I believe that will happen. Otherwise, personal medicine will not have the impact, or even worse, it's only very few people that could benefit from it, because otherwise it's going to be so expensive. When it comes to synthetic biology, I was very intrigued by the talk by Greg Venter here. But of course, personalized medicine is not only for pharmaceuticals and things like that. I also believe that synthetic biology is going to be very important for sustainability from green, beautiful organisms. I'm a photosynthesis researcher, so now I'm coming into my pet area. And I also would say, you know, these two challenges, sustainability and healthcare, there is not a Berlin Wall between these. You could say that one of the most common diseases are waterborne diseases. And also clean water, which is another area where NTU is super big in, also, of course, relates to our medical school. However, I think that maybe the next 10 years, I still believe neuroscience still hasn't had its big breakthrough. What memory or all functions we have in our biological computer here. And I always say that maybe that is the molecular biology of the soul, and this is still have to come.
So how will future pharma look? You mentioned neurochemistry and neuroscience. The big pharma in Sweden, AstraZeneca, has just fired, I think, 1,000 people, mainly in this area, because they judged that it was not economically or strategically the best way to go.
I think sorry, just very quickly, I think large pharma will be much smaller than it is now. These large conglomerates are not capable of producing the pipeline that is necessary. AstraZeneca is just going through its 10,000 changes. Pfizer have already done it. GSK did it. So I think the first thing is they're going to be smaller, because the one thing that companies do not have is a deep and fundamental understanding of the molecular basis of disease. They're chemistry development companies. They don't understand the disease.
Yeah. I wonder if there's something to be learned. I agree, these dinosaurs will probably die. We will have a small or intermediate industries. But is there something to be learned by the way these big pharmas have been managed? We all know the big management structure and the accumulation of economists and politicians in the top. In Singapore, you have another political system, I would say. You have quite a high concentration of academically educated people among the people in this deciding position. Will that affect how the development will be? Because you mentioned the gap between the politicians and knowledge, which I think is very dangerous. When this gap increases too much, there will be a reaction from the politicians to defend their positions, and they will take negative decisions, wrong decisions. I think Bertil knows the politicians.
I think in Singapore, compared to Europe, which is easy for me to compare both from the national Swedish side and Commission, the politicians here knows much more. Yeah. Would I say they have a more academic background, and therefore the gap, I think, is smaller. The risk, I think, with the Singaporean system is that they sometimes confuses speed with efficiency, and everything goes so fast. I mean, if it doesn't give results in 2 years, they close it down. So that may be the danger here. And the danger in Europe is maybe it's go too slow, so you have to find a balance.
Tom Brown, you are head of a small industry that has interesting biochemical applications too.
So yeah, I've been involved in a number of startup biotech / chemistry companies, and I mean the big pharma, as Steve says, I mean they are big. They've become dinosaurs, but there's a fantastic regeneration of small companies. I think maybe there's a lot more small companies being generated today than there were 10, 15, 20 years ago. And so the ideas are coming through, and of course the big pharma are trying to buy these small companies up and buy the ideas in. So I think this is maybe just a natural process whereby things will settle down and new companies will emerge, things will change. So I don't think the situation with the pharma maybe doesn't matter so much in some respects, you know. A natural process is occurring, and ideas are being generated by individual people. And, you know, you take Singapore, for example, you look at some of these big American universities, there's a tremendous push for people to get involved, you know, get involved in wealth creation as well as research. Of course, the danger is that we can't do everything. We are, if we want to be researchers, we can't also be businessmen, but, you know, so it's not straightforward, I would say. Yeah.
I think the comment I'd like to add with regard to pharma and biotech, having started a number of companies myself, is that it's the model to innovation. I think I completely agree with Steve's view that the days of big pharma in the context of blockbuster drugs, the billion-dollar drugs, are going to change as medicine changes and the way it becomes more stratified. However, I think there is going to be a challenge in terms of how innovative biotech companies would sort of be able to go as far as they need to go, and where the regulatory interface would come in a more pragmatic way. So I think pharma would continue, at least the way I see in the next several years, but their business model and the way they operate would significantly need to change to not only address their own survival, but to really bring in a lot of these newer ideas where it's largely driven by a lack of knowledge, but one that is going to be needed from the point of view of survival. But I think the other interesting thing, if you really look at, is this is a sort of a management study that was done in several other industries, particularly also even done at NIH and NIH Research, is that when you reach a particular stage in terms of a number of people you have, your productivity really goes down. And the more a larger a group you get in a particular division focusing on a particular area, you begin to see that you do get fairly heavy. As a consequence, your ability to be flexible and mobile is a lot less. And one of the reasons why a small company or a biotech is often successful is they're very agile, they're flexible, they're able to rapidly adapt to the challenges that they see. So I think it's going to be a combination of types of innovation, the problems that we solve, the fact that they're not going to be these big blockbuster drugs, but how do we really fill the gaps from the point of view of the true regulatory challenges we may have? Because as science becomes more complex as we understand these systems, at the end of the day, to me, it's how we balance risk. because there's going to be more risk, and regulatory sciences by definition is how you mitigate risk or balance risk. And those are set of the things that needs to be aligned in some fashion. And I generally believe that in the next 10 years, it's going to be a very exciting time with these huge advances, not only in science, but technologies that could be deployed for stratified medicine. And I think the future does look very exciting.
Could you share with us the secret of managing such a big research institution that you are managing?
Oh, that's I don't think there's any secret on that, and I think everybody goes through various organizations. I think the uniqueness of the Harvard-MIT Division of Health Sciences and Technology is the fact that it's not just one institution; it's dealing with two institutions with two very different cultures, almost poles apart, and with 200 faculty and 400 students in 8 different programs. Both institutions have come to the recognition that the key question that Harvard and MIT need to ask right now is, where do we prioritize? And needless to say, we have recognized the fact that educating people at this interface of what Harvard Medical School can do, which is training physicians, and what MIT can do, which is train engineers, and at that interface, to the point that you had made, new curriculum for medical students. This is one of the things that we have spent a lot of time making this as a quantitative medical curriculum, is what has stood the test of time. So I think we're actually in the process of going small in trying to really prioritize in key areas that we really want to have the most impact.
Yes.
How did Paul Janssen do it, huh? It's okay? Yeah, how did Paul Janssen do it? I mean, simple molecules, heterocycles. You know what happened in our part of the world? The United States had a lot of money and time after the war, and we got R.B. Woodward to teach us how to get over the deserts and into the wilderness of box canyons, had ski areas, all kinds of stuff. We made molecules by 100 steps, and we were proud of it. I mean, this really is where these guys are on the 40th floor now. It's totally dominant in the big pharma, and then comes all these other methods of discovering drugs, proteomics, genomics. Hey, you need one molecule. That's all. Or you need a few. You need the function. You don't need to know why. People will study the why of the molecule that works until the cows come home, right? That's what we do. That's what humans do. But it's not the way you find things. You can't. And everybody thinks their method's going to get the new drug, and they're going to be, even the guys in the chemistry thing are misguided in that sense. You got to really be cruel to. I think the blood would flow. These guys that run the big top floor, they don't care who gets. If there's a new method to get one quick. See, quick seems to connote dangerous. Quick is no, I'm saying quick, the best drug. I would like to have a shot at a target for a couple of years. That was really important. And if I couldn't get, I wouldn't want any salary unless I got it. But I need some chemists and a good screeners. I need screeners that really screen. I mean, you got anybody who has a real live animal squiggling under the thing and sees suddenly float, changes on blood sugar, and he just monitor. You don't have to do anything until there's an excursion outside the set points on your machine, right? He just has a little stuff going into him, and you can even use them over. It's wasteful to throw the animals away because they already had one run of something, right? Because you can always go back. If it's quick, I can't get over how much they want to screen the monster libraries. They all are very much the same. They got piperazines for 15%. Of course they do. That's made from ethylene, ammonia, and chlorine, and it's not a natural product even, but it's for sacred pharmacophore. And their library will have it as a connector. So they don't throw it out. They get the hit and they change it, right?
Barry, I just got an idea. Maybe we should change the policy. Everybody seems to agree about the environment, the carbon dioxide, that we should have a tax on that. Our health is something that concerns all of us. But today it's the drugs. They are driven by the market. Nothing is made for malaria, for instance, because that will not pay back.
That's embarrassing, isn't it?
So maybe the nations could go together in the same way as we did for the environment and negotiate about how much...
Andreas has an idea. He told me yesterday, it was interesting. You want to tell him about your idea? I think third stage clinical trials should be small, and they should watch it like a hawk. It should not be necessary, and then they'll get all kinds of benefits out of that. It'll be kind of like seeing the other aspects, right? And Peter Lansbury, I think it's a great idea, but of course it just makes the lawyers start salivating, I guess. I mean, you know, pharma wanted to have complete release. That's why they get the $1 billion. They don't want to touch it after it's released. That's not right. You know, that's just not right.
We could discuss that later at the dinner. I think myself that it was a good idea. Maybe that could be the topic of the next Molecular Frontiers Symposium, which could then also have a sort of contract where all the big nations at least sign, a Tokyo agreement or something. What do you think? I think I would like to ask, I see some of you look, yes, one question from the floor.
Hi, I'm Kim Ho from Ministry of Education, Singapore. I thought I'd ask a question that may have some impact on the young people among the audience over here today. I hear about, talk about maybe relooking at medical education. What about relooking at even the way that our students learn science? Craig Venter touched on it yesterday, and I think he related that he was fortunate that he wasn't turned off by science at a very young age in school. So my question is actually, we've been actually training, getting our teachers who have been trained in the various universities in the very traditional disciplines of physics, chemistry, biology, and they bring that discipline into the schools in very much the same way that they've been taught by their own lecturers back in university. How can we look at the whole structure of maybe the sciences, even in university, even in school curriculum of physics, chemistry, biology, to maybe engage our young people in learning science in a very different way?
I think we heard some answers to that it's your school teacher that primes you, but then you may have a lag phase. You may do something else, and then you go on to it. And there is a big stochastic factor, I'm sure. There are all kinds of good jobs to be taken. I would like now to ask our president here to round up this.
I think that question, if I may answer that, I think the science teaching in Singapore is quite good. It may be a little bit conventional, and I think one needs to combine the disciplinary thing with the more lateral thinking. So I think Singapore could actually be a role model, because I think you are closer to fixing it than most of the countries I know of in Europe. So I think you should be. I know you are thinking and are concerned about to improve it, but you are not in the worst position. That's my thinking. I can tell you countries that are more worse. So I have enjoyed...
Sorry, I won't let that go unanswered. I think there is more to it. I think the teacher at school and the role as a mentor is very important. And we have sort of a clash here. We have the high quality of the students. The students are driven to deliver all the time. It might be a little risky. You need also the human ingredients. And I think several of you I've heard now from, including Craig Venter, that you need to disconnect at some period in life. And that is a little in conflict with the streamlined distillation to get the top student. I think if you are aware of this, you could include it in the system. And I think very much happens in school and in the high school and even before in the inspiration.